like this. I'm just going to minimize all so I see the screen. Okay, hi. So my name is Joran Deschamps. I'm coordinating a small team of software engineers that is shared between the UG Lab in the Human Technopole and the National Facility for Image Analysis. And we are also a member of AI for Life, which you already heard from. And so today I'm gonna, I'll have to click like this. So as hinted before, I'm going to talk about a specific type of image restoration, which is denoising. So denoising, I think everybody's familiar with what noise is when you have data. In microscopy, it ends up looking like this, where if you have a strong amount of noise and you have very low signal to noise ratio, then you get images where downstream quantification is very difficult. And the task of denoising is to try to clean up the amount of noise from each pixel so that you get something that you can actually, for instance, segment later on. And so I'm going to talk about two tools and then a bit how to use one of them and a bit the limitation towards the end. And I'm going to try to echo as much as possible to what you heard already last week and what Danielle or the other speaker this morning talked about. Okay, so the first tool is called CARE, so Content-Aware Image Restoration. It was published back when Florian and collaborators were in Dresden in 2018, and this was one of the first big efforts in bioimages to use deep learning networks in a scientific manner for image restoration. And so the principle you already seen last week, but you basically have a noisy input to which that you feed into a neural network, and you compare this with ground truth. So you need in this case, it's a supervised approach. So you need to have already acquired a ground truth reference image that is paired with your input. And you compare the output of the network to the ground truth. The value, well, you get an error out of this, which is usually called a loss, and you backpropagate this loss to tune all the parameters of the network so that you minimize this error. And you do this for many, many, many images during training. And if you did a good job, at the end, you hope that you end up with an output from the network that is very close to what the ground truth is. Well, I can't show the video, but let's try to have it in the screen here. So at least you get a little bit of tease. So it works really well. Now, in the paper, there was a lot of comparison with classical denoising algorithms. So not deep learning, and it vastly outperformed them if you compare the deep learning output compared to the ground truth. Now, Daniel already demonstrated one interesting approach in CARE, where you look at you shift the dimensions to try to get a better resolution. They also did some interesting things like not going from signal to noise ratio to low SNR to high SNR, but actually going from 3D low SNR to a denoise projection. So just to give you a bit of an impression of the diversity of approaches that you can have in that case. What if you have no ground truth? So this is a problem that you will find a lot in biology and in microscopy, is that you can have, for instance, fast processes where it is not possible to arrest the images. If you have a longer exposure, then you will miss all these events because they will just be totally smeared. This is, I can also try to show it here, where you distinguish some peaks, but actually you're missing a lot of the peaks. And another way is, for instance, when you're imaging, just simply destroy your sample. So what you only get is a single image. You cannot acquire a ground truth from it. So that's one of the main reasons where one of the main roadblock to use CARE. The other one is the amount of ground truth that is necessary. So if getting one ground truth image takes weeks, it's going to be difficult to build a data set such as what Daniel described. And so that's where the idea of noise to void comes from, trying to solve this problem for life scientists. And so it came from, again, back when the lab was in Dresden and the MPI CBG. And the idea there is you don't have a ground truth. So let's teach the network to guess what single pixel value is. Now, the problem is that if you keep this pixel value in the input, it's very easy for the network to know what the pixel value is. So what we do is mask in the input certain pixels for efficiency, a certain number of them, and then we train the network just on this one image, on this one corpus of images, to estimate what the pixel value is from the surrounding pixels. So that's a bit of a strange idea, a bit of a departure from the common deep learning scheme. If you consider that a noisy image, so each pixel has a component that is signal and a component that is noise, then noise to void will potentially work under two key assumptions. And the first one is that the signal is continuous. So if you have a certain structure and you're missing one pixel, then it should be quite easy for you from the surrounding pixel to guess, to have a pretty good estimate for what the amount of signal is. In biology, the structures are continuous. The microscopy does not break this. So this is easy to uphold. Now, the other assumption, and this one is very key to noise to void working, is that the noise is independent from one pixel to the other. So noise is pixel-wise independent. What does that mean? So for instance, if I throw a coin in the air and I look at what is the probability to get a tail, this probability is 0.5. If I throw the coin a second time, then the probability to get a tail is totally independent from the first throw, from the outcome of the first throw. And so this is in this assumption here, this is the same thing, same mechanism, is that the noise, the amount of noise in one pixel is independent from the amount of noise in the other pixels. This is in general true for a lot of microscopy methods. In microscopy methods where the main component of the noise is the shot noise, so it's Poisson distributed noise that comes from the particle aspect of light, then the amount of arrival times that you will get, the number of photons that will arrive during one exposure time will be independent from the amount of photons that you get in the next pixels. And we'll come back to this. So when those two assumptions hold, then noise to void works wonderfully well. And remember that here we're training without ground truth, we're just taking one image. As long as the image is big enough, we mask enough pixels, we just train. And noise to void train really, really well. So it trains fast to a point where you might get away with not having a GPU for this. It works also very well for fast processes. So I'm sorry for jumping back and forth. So if we look at the same type of data, then you see that where here we start in the denoised images, we start seeing, and I don't know if you see the cursor, but we start seeing a lot of peaks that are almost imperceptible because they're drowned in the noise. They are there statistically, but they're difficult to see by the naked eye. And the picture on the right is what we call the residuals, which is an interesting control for noise to void, which is the difference between the input on the left and the denoised data that is the output of noise to void. And if you don't see any structure in there, that means that noise to void effectively only remove noise. And so that's a good quality control. Now, you might argue that, oh, but I do see the outline of the cells. And the reason is, if you're in a shot noise regime, where shot noise, the Poisson distributed shot noise, is the main component of your noise, then the amount of noise that you have, the standard deviation, scales with the intensity. So regions that have a higher intensity will also have noise. So the difference between the mean of the distribution and the values that you find will be, in average, much bigger. So the more intensity you have in your images, the more noise you have. The signal to noise ratio still being higher. So this is a really good control for seeing whether noise to void actually worked. Now coming back to this, I want to insist again on the difference between a supervised approach like CARE and the self-supervised approach like noise to void. So this echoes what Daniel was saying before, is that you have a training set that is actually quite an effort to acquire because you need pairs of raw and ground truth images to train CARE. You need to have validation images to check throughout your training that your network generalizes to unseen images. And because when you are happy with your training, because you stop the training based on the criteria that it performs well on your validation images, then you cannot use the performances on the validation images to report how good your performances are. And that's why you need the test images. So images that have never been used throughout the training. And so that's when you publish something with deep learning, that's what you want to report numbers on the test images. And now after that, well, as long as you take raw images that are within this distribution of training images, then you can predict on those. And I insist again on what Daniel was saying before, is that if you then feed for during prediction images that are not similar to the training images, then you're going to have hallucinations. For noise-to-void, we do not have ground truth. And therefore, we only train on the raw images. We mask the pixels. Validation images can be used to check that it does not overfit. Usually, we either take them directly from the training images, so you cut a part that is not, that you extract from the training images. It's not anymore in training images. You just use it as validation. Or you can, if you have a bunch of images, you just take some of them for validation. Now, we don't have test images. There is no number we can quantify at this stage. And you can just predict on your training images. And because noise-to-void trains very easily and quite rapidly, this is not the type of network that you usually keep on the side to predict at a later time. You usually retrain it every time you have an experiment. So you trained on images, predict, keep both raw and predicted images. But so how to know that you're doing it right? So the first thing is to check the residuals for structures, as I showed you before. Another way is like in any type of experiments, especially in biology, you want to have controls. So you don't trust your gel. You have a scale to make sure that what you're looking at makes sense. And so one way to do this with noise-to-void is, for instance, train multiple networks and compare the results. See a bit where the networks will give you different structures or different results. You can also train on 2 different sets of images and then cross-check the predictions on similar images. And then when it comes to publication, if you can generate ground truth for at least a subset of the images, then you can also make some quantification. How do you use noise-to-void? So in a scale from, if you go from low expertise in deep learning and Python to being more of a Python expert, the first thing is that there is a noise-to-void Fiji plugin. Now there's a bit of an asterisk there because it is based on quite old technology, so we are not really capable of maintaining it anymore. So you might have some problems in the setup to manage to get GPU training or even to get it to run. We have good hopes with current work at the Pasteur Institute, Madrid and Wisconsin, from the people behind Fiji maintainers, to be able to retrain soon this type of network in Fiji and ImageJ. But currently, we're not really advising this. Now, ST mentioned last week the zero-cost notebooks that allow you to use Google resources to train. So there is noise-to-void, there's CARE. You also have more recently ST's work on DLMIG for everywhere, where you have Docker containers, can run it locally and also on some servers. And then what we usually advise and what we use when we need to denoise images is the NAPARI noise-to-void plugin. I will talk a little bit more about this one. And if you're more comfortable with Python, then you can use the Jupyter notebooks from the noise-to-void repository, or simply call it in your own applications as a package. Okay, so NAPARI, if you're not familiar with it, it is a multidimensional image viewer in Python, and because most deep learning is done in Python, then it's very easy to interface with those algorithms, as opposed to Fiji, which is in deep learning, in Java, sorry. So the NAPARI plugin is what you see on the right, and I again need to go to an animation. So what we see when it runs is that you have linked to the documentation, to the GitHub. There's a little indicator that tells you whether it has access to a GPU. And then you can train either from the layers, the images that are loaded already in Napari, or you can train from the disk. You have a bunch of parameters, which are kind of the parameter that you usually would change. And if you're feeling a bit more adventurous, you also have more advanced parameter to change the architecture. Once you trained, you have some progress bars that show you how far along your training is. So the steps are something that constitute an epoch, and at the end of each epoch, there is the loss. So this average error that was calculated on the training set is plotted. And so this plot kind of shows you how well the training is going. Usually you expect noise to void to learn the structures in your image very rapidly, and then plateau for a long time. And this plateau actually, you have to remember that the loss, because we're training the network to guess what the noisy pixel value is, which it cannot because the noise is independent. Therefore, the loss being fluctuating but kind of stable just means that the network might still be learning to get closer to the real, to its estimate of the real value, but it will never go to zero because the zero would be the noisy pixel, and that's not what we want. So usually noise to void looks like training looks like this. You have this sharp decrease in the loss because it learns all the structure, and then this regime where it fluctuates a little bit. And usually we try to let it run for a bit, then predict, see if we're happy with the result, and then either retrain or keep it that way. So if you want to retrain, the Napari plugin allows you to reset the model from 0 or to continue training to train a little bit longer. Now this is a big image. Doesn't look like this, but it's probably 2000 by 2000. You just predict tiled, and then you should see the images that are already shown in this presentation that has been denoised, and the prediction is quite fast. Okay, so now something that is very important to discuss, as always, is what are the limitations of these algorithms. And many of the most important points have been raised already last week and this week. One particularity, though, of noise to void is that you can have checkboards artifact, which is this little crosses that you see indicated here by the first red arrow. We published recently, so Tim Oliver, who was one of the author of Noise to Void back in Dresden and collaborator from Zeiss, Noise to Void 2, which by a small modification of the architecture gets rid of this of these small artifacts. Noise to Void 2 is available in the Napari plugin, but only in 2D, and we're working on the 3D version. So we talk a lot about uncertainty, which is difficult to assess when you have a network like Noise to Void, because Noise to Void is deterministic once it's trained. If you pass as input the same image multiple times through a trained Noise to Void network, you will always get the same output. And this output is basically kind of the average of all the possible images that corresponds to the low signal to noise ratio image. Now, still back in Dresden, we published a Denoising, which has a slightly different architecture, which allows you to sample this distribution. So to draw stochastically different interpretation of the low signal to noise ratio image. And this is important because it allows you to see which portion of the image are stable, which portion of the image the network is pretty sure of itself, which doesn't mean you should trust it 100%. It means that the network will not give you any variability, but you should always have control. Now, what is interesting in there is that it can show you where the network is very uncertain. And these areas you should definitely not trust, because the amount of information that was in the low signal to noise ratio images is just not sufficient to say something to say something specific about the structures in there. So this is important. A big limitation of another big limitation of noise to void is when you encounter structured noise. So I mentioned this is one of the fundamental key assumption of noise to void is that the noise is independent from one pixel to the other. The amount of noise in one pixel is not influenced, not correlated to the amount of noise in the surrounding pixels. Now, if you do violate this assumption, if you have structure in your noise, so we're not talking about a pattern that is from the, let's say, fluorescence of your sample, but we're talking about really the amount of noise from one pixel to the other. So if you here it's artificially added to an image, so you have a bit of structure in the noise. If you train CARE, right? CARE is a supervised approach. So if you train CARE to get rid of this specific pattern, then it will manage to do this. Noise to void, on the other hand, because its assumption is that only the signal is continuous, then it will consider that the structure in the noise is actually signal. And then it will do the same thing that it does for the signal. It will just clean it. Right? The structure that you see is in the data. It's not inventing it. It's just cleaning it and therefore making it more visually striking. You do encounter this type of situation in microscopy, especially when you're in point scanning methods. So, and this is a result that you get, for instance, with noise to void, where you see that this pattern then appears clearly. Now, I will insist this pattern is in the data. If you take the 3-photon mouse image that is at the center here, and you have a time lapse and you average it, then you will see this type of structures in the data. Right, so noise to void also serve as a detector for this type of structures. And so more recently, we also published a new algorithm. It's called HDN for hierarchical denoising, which is a bit more of a complex network. But what it allows is to separate this structured noise from the signal. And so this works quite well. And a lot of our current efforts is to trying to make all those algorithms as available to the community as noise to void is. So unfortunately, especially the PPN2V, denoising, HDN, those algorithms are very difficult to use because they're still in a research code state. And we're now trying to put them into one neat package. So this is very much work in progress, but hopefully you will hear from that very soon this year, or next year rather. Okay. So a bit of a final word on CARE and noise to void. So just to remind you, CARE requires ground truth. It's supervised training. That means that you need to have a certain effort building the training data sets, because this training data set must sample all visual features of interest. If you do not have this, then you run the risk of having a bit of hallucination. So you always need to be careful and explore your data. Noise to void does not require ground truth, and therefore it's a very good first step in denoising, seeing what comes out. It can have a bit of checkboard artifacts, so don't be surprised if you see it, and it will clean structured noise as well. So you need to be aware of this. I mentioned already how to try to have a bit more scientific approach to noise to void, and how to be able to control whether what you're seeing is present in the data. So uncertainty of the network, train multiple networks. Noise to void as CARE, if you trained it on a certain image and then you apply it on vastly different images, you will also have hallucinations, because noise to void does learn structural prior about your data, what is in the data. So it's a general, what Daniel mentioned is a very general problem with deep learning, problem of hallucinations. And then intensity quantification is also a big sensitive topic, because it is not clear whether this approach maintain the same intensity for your images. So noise to void seems to be better at this than CARE in general, but it will have a tendency to lower the high intensity and increase the low intensity. So it will pull everything a bit to the mean. So the general rule is always start by doing your intensity quantification on the raw images. Always keep your raw images in any case. So to do more echo to the previous talk, should you use deep learning? Yes, scientifically, but only if you need it. It's a bit of it might seem like it brings a lot to your paper because it makes it a bit more fancy, but you run the danger of doing something wrong. And you might also spend a lot of time on this. So if you don't need it, don't use it. if you do need it, read the literature on how to best do it. There's some good nuggets of knowledge in this paper. And be sure to enable others to reproduce your results. So provide your data as much as possible openly. Give your train network and also your training routine to the community. Make sure that you share these things. And as was mentioned before, and also by ST last week, within AI for Life, this big EU grant that has like 10 partners, we're trying to make deep learning more open and more fair. And so among all the activities that are done within this consortium, there is the bioimage.io model zoo. It also comes with certain formats that brings a lot of metadata on, the traceability and the fairness of the network, but also who trained it, how, what is it supposed to be used for. So this is a very, very important step. So if you publish, please upload your train model there, and also upload your data to the bioimage archive, which is now building up to have AI-ready data sets. We also have a lot of workshops, and we will have in the future workshops also on how to use all this infrastructure that's being built by AI for Life. And the last word is we also running open calls. So every year we have you can apply with your data and with your project, and there is an independent reviewers panel that selects certain projects, and then the open call team within AI for Life just will try for a few months to improve your workflow and try to employ deep learning on your data. And so be on the lookout, because we're going to open the next open call early 2024. I think you will have until February to apply. So think about this if you feel that you need some experts to look at your data and help you develop more deep learning approaches for your project. And with this, I'd like to thank the UCLab, the AI for Life Consortium, the research software engineer team, and the team at the image analysis facility. And we're going to have a lot of position in the next years for the national facility. Thank you.
Thanks a lot, Joran, for your presentation and yes, showing what is possible, also what is still not possible, and that was super great to see all this progress on the field. And we had a question actually, at least one from the audience. So I don't know if Krista, you want to ask yourself. I cannot see if yes, you can maybe switch on your video on sound. Otherwise, I can have no audio. Okay. So you had a question regarding the noise to void version of the NAPARI version, sorry, of the noise to void plugin, I believe. And the question was how one can actually set the few parameters that you have, and yes, actually, how do you guide users in that, and how should they be changed?
Yes, so we have in the documentation, we have a description of all the parameters that need to be changed. It is, I agree, a bit daunting. In general, yeah, we have a link to the documentation, and we try to document as much as possible. In my opinion, we're still a bit light there. And one of the things that we are currently working on is also trying to make a big documentation effort for the next. So I mentioned this package we're working on. We're also doing there a lot of experiments and a lot of description of in which situation would you want to change which parameter. So this is something that is I agree, important and usually not always done super well. So as far as the NAPARI plugin goes, there is some description of all the parameters. I usually don't change them so much. Just you want to change the epochs and the steps for the length of the training, and then you want to change the patch and batch, so P and B, to set basically the memory footprint on your GPU, because this is usually the first error that everybody will encounter is you set the parameters too high, and then your GPU memory is just too small for this. But usually the more advanced parameters we do not change so much, and then there's some description and documentation on how to use them.